Hi, everyone. I want to thank the organizers for giving me the opportunity to talk to you today. My name is Paul Renner, President and CSO of Alita Biotherapeutics, a cell therapy company in Natick, Massachusetts. I won't talk too much about Alita today, just share a little data at the end. But I want to talk to you about what we see as a roadmap for successful immunotherapy, particularly for solid tumors. And I'd like to do that in the context of thinking about immunotherapy responses, resistance to immunotherapy, and the geography of immunotherapy. And we'll sort of take these on in order as we go. Now, if we start at the beginning, we would have to go all the way back to IL-2. And so let's just not do that. Let's just dial back 15 years to 2006 in a paper published by Gallon et al. In Science. It's a very interesting paper, predates the PD-1s, and is coincident with the development of CTLA-4, and talks about the density and location of immune cells with respect to outcomes in colorectal cancer. And so this is a heat map that was generated. It's transcript profiling, identifying immune determinants of patient survival really as a consequence of how quickly they relapse from standard of care therapy, and correlates that with the expression of Th1 immune responses. So we can see granzyme B here, we can see interferon gamma and signaling, we can see CD8s, all lighting up in the adaptive immune heat map. And we see some components of immunosuppression in the lower right-hand corner here, not correlated with a good outcome. Notably here we have TGF-beta, we have a hint of VEGF. CD4s are here, possibly because of Tregs in the tumor microenvironment. We'll talk about immune cell geography later on. So this is a very interesting paper, 2006. Now if we move a few years ahead, to 2010 and through 2012, we start to see the publication of the first anti-PD-1 and anti-PD-L1 trials. So we've got, the first one I could find is a phase 1 study published in the Journal of Clinical Oncology by Bramer et al. This is the Medarex anti-PD-1. And then, of course, in 2012, we had the dual publication of an anti-PD-1 and anti-PD-L1 studies in the New England Journal, showing really long responses in several studies of melanoma and non-small cell lung cancer. So this really started the snowball of activity in immunotherapy. And I think what we learned from looking at responses, right, and this was summarized by Dr. Weber in the ASCO 2018 education book, and it reflects really data coming from metastatic melanoma, but it's applicable to any number of different tumor types. A good response to checkpoint therapy is associated with the appearance of PD-L1 on tumor cells, also on myeloid cells, the appearance of CD8s in the tumor microenvironment, and a transcript profiling signature, that's reflective of immunity, and perhaps also, of course, tumor mutational burden, depending on the cancer type. Not so important, perhaps, in melanoma. Okay, so these types of studies and these sorts of reflections on the field led us to develop the broad concept of cancer immunity, made famous in 2013 by the paper by Dan Chen and Ira Mellman, published in Immunity, called Oncology Meets Immunology, the Cancer Immunity Cycle. And this cycle, right, which starts with tumor cell death and the transport of antigens to lymph nodes, stimulation of T cell activity, and then trafficking back into the tumor in order to engage an antigen that the T cell population has perceived. This concept has stood the test of time. One of the implications of this cycle is that there are lots of places to intervene. As you go around the circle, there are lots of positive regulators shown in green and negative regulators shown in red. And I would argue that despite all of that, we've mainly focused down here in the corner, right, where the tumor is. Much of the activity has really been focused on the tumor microenvironment. Nonetheless, diverse opportunities to intervene if one goes after, one or more of these additional targets. Additional meaning not CTLA-4 or PD-1. All right, so, not to miss a beat, we played off of this concept in 2015 in a review we wrote for Nature Reviews Drug Discovery. This is just one of many figures in that paper illustrating the proliferation of targets. This particular figure has antigen-presenting cells on one side and T cells on the other side, and shows you an array of activation moieties that could be targeted by agonists and checkpoints that putatively could be targeted by antagonists. And of course, the idea here was that there was a rich field of possible immune checkpoints and immune activators that we could go after to improve anti-tumor immunity. So, the field at that time was dominated by this idea that we just needed the right combinations of antagonists and agonists for any particular indication in order to boost the anti-PDX, PD-1, PD-L1 response. Famously, summarized as releasing the brakes and stepping on the gas. But as we all know, we never say this anymore because it really wasn't that simple. And the reason was that resistance to checkpoint therapy is complicated. So this is a paper I really like from Cell in 2016, Hugo et al. And again, the title is reflecting, analysis of metastatic melanoma and then pathways of resistance to anti-PD-1 therapy in metastatic melanoma. And what pops out is a big VEGF signal, a TGF-beta signal, an epithelial mesenchymal transformation signal, also hypoxia. So, it's a complicated array of pathways, but it boils down in this instance to, three things really: VEGF, TGF-beta, and not shown in this figure, but in another figure in that paper, beta-catenin. And a number of years ago, I gave a series of talks on this type of data, referring to these pathways as the big sticks of immuno-oncology resistance. So this signature, which has now been called the innate anti-PD-1 resistance signature, as I said, the signature was articulated in that paper in melanoma, but if you look carefully at that paper, you'll see that the signature is reproduced in lung, colon, kidney, and pancreatic to different degrees, and the degrees are reflected by the amount of blue versus red, across these indications. Nonetheless, this iPress signature, appears in a number of cancer types, suggesting that it's a common way for tumors to resist immunotherapy, and of course associated with outcomes. So if you're enriched for the iPress signature of resistance, your response to therapy is worse, and your outcomes are worse than if you're not enriched. So that makes a certain amount of sense. Now, this resistance literature, right, was rolled up into another review in Immunity in 2019 by Hegde and Chen, and really articulated this idea that there were a number of different immune phenotypes that were imposed by the immunosuppressive signals. So we're familiar with the inflamed, that's where the PD-1s work. The immune excluded, where the lymphocytes are kept out of the body of the tumor by a variety of signals dominated by TGF-beta and VEGF. And then immune deserts that can be set up by any number of signaling moieties, where there really are very few CD8 positive T cells at all. And this was highlighted as one of the 10 top challenges in cancer immunotherapy in that paper. So one of the things to consider is that these discussions on response and resistance reflect the tumor microenvironment geography, right? Because that the T cell biology that we're assessing in the tumor is effector phase, right? So that's downstream of activation. And what we're seeing there is that positive activity is associated with CD8s, interferon gamma, granzymes, et cetera. And the tumor cell biology is, of course, tumor microenvironment intrinsic. It's set up by the tumor cells. So here we see the resistance signature and some of the more common other modes of resistance, like loss of HLA expression, loss of responsiveness to interferon, and other mechanisms of immunosuppression. So this is a tumor microenvironment centric geography that we've been discussing. So, you can sum all that up and make it pretty simple by saying, well, there's one way to trigger a response, which is to get CD8s in, and there's lots of ways to block it. So let's go back to the response part and just ask the question, how does the response get triggered? Where does it come from, and how is it sustained for the benefit of the patient? So again, right, the tumor undergoes some cell death, antigens traffic to the lymph nodes, and so on, right? So these are dendritic cells that do the heavy lifting here. CDC1s, they go by a variety of names, have been articulated by a lot of labs as being critical. And they go to the lymph nodes and they trigger anti-tumor immunity, and then of course we have the rest of the cycle as I've already described. Now, one formal question is, how do you know the lymph nodes are important? And, that's been answered in animal models in a number of studies, two of which are shown here, which just say that if you block PD-1 in the draining lymph node, it's sufficient to trigger anti-tumor immunity, right? So that makes sense. So we know that the lymphoid organs are critical here, and we can interfere at that level to stimulate tumor rejection in animal models. Okay, now let's add a shortcut, right? So we've got this 7-step cycle here, but now we know that there's a shortcut whereby a lymph node structure can actually be induced to form in the tumor itself, and that opens up another door. So these little lymph nodes, also known as tertiary lymphoid structures or tertiary lymphoid organs, were described as being related to outcome in response to immune checkpoint therapy in a bunch of papers in late 2019 and 2020, including these 3 published in the same issue of Nature in January 2020, showing the association of these CD20-rich organized lymphoid structures in sarcoma, melanoma, renal cell, and other melanoma study. In other words, the presence of these in the tumor were associated with a positive response to immune checkpoint therapy. So that is a super interesting observation that I think is often overlooked. So let's just dig into it a little bit further. So, there's this notion that there are a lot of B cells there, right? You see it in the graph, you see it in the stains. What are the B cells doing? Because we just said that it's these dendritic cells that are trafficking out of the tumor and carrying antigen that are important. So, why do we need these B cells? And the answer is that it's not just the B cells, it's the role of B cells in setting up the immune geography in these lymphoid organs. So here's a lymph node. This is stained rather beautifully with a B cell marker showing you the B cell follicles. There's a T cell zone. This magenta color or pink color is the lymphatic vessels. So that's how the dendritic cells get in. And then these cells traffic through the different zones in a somewhat complicated dance that's been articulated heavily in the literature. So you can go read about that, but it's very cool. And it's duplicated in the tertiary lymphoid structures. So here we've got our B cell follicles in red, and we've got a T cell zone here in green. And these authors, Lin et al. In the Frontiers in Immunology a couple of years ago, provided a nice cartoon of what's going on here. So the B cells, in collaboration with follicular dendritic cells, set up the B cell zone. The T cell zone is adjacent to it, contains T cells and dendritic cells. The entire thing being orchestrated by chemokines and so on and so forth. And this structure is meant to foster, the development of immune responses, right? That's what it's evolved to do. So why is the structure important? And in particular, why is it important for CD8s? So FDCs and B cells organize the structure. This allows the famous immune triad to form between T cells, B cells, and dendritic cells. We know from other literature that CD4 help is essential for CD8 T cell population differentiation, entire thing being controlled, orchestrated by chemokines, supported by signals coming from cytokines and co-stimulatory signals. You get the idea, these cells have to come together in order to induce an immune response. What's really interesting is that for CD8 positive T cell differentiation into effector and memory cell populations, a sustained interaction or embrace between the T cell and an antigen presenting cell like a dendritic cell or a B cell is required. And that embrace is regulated by adhesion molecules. So I've listed a few here that are of interest. There's a lot of data now on LFA1 ICAM interaction. And so here's the expression of ICAM on a B cell. And what you can see is that the ICAM overlays and rings the B cell receptor. And that's the point of contact, of course, with the T cell. Not only that, the expression is upregulated and sustained in this graph through 40 minutes, and the description of this kind of upregulation and interaction of a T cell with a B cell or a dendritic cell extends out to several hours in some papers. So this is a organizational structure and an interaction of the lymphoid cells that isn't duplicated in the solid tumor microenvironment unless there's a TLS there, right? This isn't going to happen for T cells that are just parked in the TME. There's no organization there. Okay, so let's keep going. Now, so those are a bunch of observations about what might be happening in the lymphoid structures that's useful for supporting T cell immunity. So are these observations actually useful? And I'll give you a couple of quick examples. One from our work published earlier this year showing that a CAR T cell that can see a B cell, even if it's a lymphoma, is much better off in terms of fitness and proliferative extent than the same CAR T cell that sees a HER2 positive carcinoma. So we've engineered a CAR that can see either antigen, and we put them in a restimulation experiment. And if they're restimulated with a HER2 positive carcinoma, you get a little bit of proliferative increase through 4 rounds of restimulation. If those same CARs are stimulated with a B cell, presumably having the adhesion benefit, among other things, you see a 100-fold increase in terms of cell number after 4 rounds of restimulation in about 12, 14 days. CAR T cell therapy is a numbers game, so the more you get, the better off you are. This may explain why CARs to CD19 and to a certain extent to multiple myeloma cells, so B cells and myeloid cells, do rather well, while CARs to other antigens do rather poorly. So that's one example. Here's another example that I really find fascinating. So this is 2 labs. One is Darryl Irvine's lab at MIT. This was published a couple years ago in Science, where they've coupled antigen to albumin, sent it to draining lymph nodes where it's engulfed and presented to CAR T cells to extraordinary benefit in their animal system. So that's just saying that if you provide an immunologically relevant depot of antigen for a CAR T cell, the outcome is better. So that makes sense, right? Another example, and this is really cool because this is the technology that BioNTech used to make the COVID vaccine, but it was being developed as a vaccination strategy to support CAR T cells. So this is the RNA vaccine that's delivered to lymph nodes in order to support the efficacy of CAR T cells. Redeployed for the benefit of antiviral immunity to create the COVID vaccine. So, these are rather interesting papers. I just find this stuff really cool, and it makes immunological sense. Okay, so I want to leave you with a question. This is HER2 staining in a HER2 positive breast cancer biopsy, and as you can see, it's really intense, right? And expression levels of HER2 can reach millions of copies per cell on the cell surface. A lot of antigen. And yet, anti-HER2 CAR T cells don't expand very well. They don't persist very well. They have minimal efficacy. Dr. Ahmed's work in sarcoma aside, he has had one or two patients do okay, but in general, these strategies have been quite unsuccessful. And I'll leave you with the question of why is that, right? Why don't CARs to HER2, a solid tumor antigen, work well? And I'll just invite you to stay tuned for the answer in an upcoming talk. So let me end by acknowledging the folks who contributed the data. Roy Lobb, co-founder of the company, our SAB members across cell therapy centers in the US, and our support from Cancer Research UK. And I will stop there and say thank you. You can always reach me easily by email or via social media. And I will stop there and say so long.